I'd like to talk about oyster biology, as everything that you do with land-based oyster farming will revolve around enhancing the oyster's biology. So it's a very, very simple mollusk, bivalve mollusk. However, to get it to grow, you've really got to understand it. A great place to start in that process is Goldstoff, 1964. I would recommend that for everybody. It's just worth a read. The work that was done way back then is absolutely brilliant. The main species involved in commercial aquaculture today are Crassostria virginica, the US, the western coast, sorry, the eastern coast American species, which is very, very similar to Australia's Sydney rock oyster, Sarcostria commercialis. The other one that you would all be aware of is the Pacific oyster, Crassostria gigas. It's a faster growing, larger species, cold water basically, and this has a lot to do with why it's so successful in Tasmania. The other species that should get a mention here is the sleeping, what I would like to call the sleeping giant, and that is the Queensland blacklip oyster, which is basically a tropical version of the Sydney rock oyster. It doesn't suffer from the disease issues associated with Sydney rocks, and the Queensland coast is absolutely prolific. Everywhere you go, you will see this oyster. People eat it. It is very nice. Matter of fact, it's hard to tell the difference between the taste of the Queensland blacklip and the Sydney rock oyster. You can tell, of course, because the edges of it show a distinct black edge, consequently the black lip. In simple terms, oyster biology, we're dealing with a bivalve oyster. The part by which it hinges is called the hinge. The muscle that holds that hinge and the two bivalves together is the adductor muscle. It surrounds a fleshy component of the oyster itself, with the structure off to one side incorporating the gills, and the area which is of a creamy colour is the gonads, and the body here is the intestines. So you have an anus here and a mouth sort of attached to the gills here. Okay, that's basically it. Really, that's all you need to know. You have a frilly edge around the what they call the mantle edge. So the oyster has a thin skin layer which travels out here, and it's the edge of that mantle layer which creates calcium for the shell. So this is the side of growth, continual growth. It's around the outside of the shell. So an oyster starts out as a blob and then grows exponentially like this, so that at 3 months old it would be something like that, and then it moves out basically like that. So market size across there would be 75 to 100 millimeters. Let's call it 75 millimeters is the perfect oyster, and that's how it grows. It just extends that shell out over time. Right, so a Sydney rock oyster, its mouth limits the size of food that it can take. So you're roughly looking at 25 microns is the maximum size of food that this animal can ingest. It does take larger particles in through its gills, so it catches its food by pumping water across these gills. The gills have ciliated hairs on them which collect the food towards the back of the gills and then transfer it to the mouth. It passes through the system and then out the other end. The food that is too large comes out here, can be seen as a mucus-bound filament of what is called pseudo-feces. So having understood what an oyster needs, it's easy to take it to the next step and sort of look at a simple design for large-scale culture. You know what the issues are in the wild, you know what the issues are with trade culture and normal ranching of oysters, which has been around for a few hundred years. So, therein lies the idea of a tank. We take a tank, in my case a 100,000 litre tank, add the water and give it an environment that will create optimum growth. And that's exactly what land-based oyster culture did. What was remarkable with land-based oysters was the density which you can actually grow them at. It's really quite amazing. It's like systems used in hatcheries where they use basically fluid tanks, so the oysters are in constant suspension, then a millimetre in size at that point in time, and they are kept in suspension as a way to grow them. It works really quite well as long as you stay within the parameters. You need the feed of the right size. You need the flow rate to be enough to supply oxygen and feed without rumbling the oysters and killing them. Same with this system. You just got to look at a land-based culture system and apply the simple rules. We need to supply food. We've got our biology. We know what the oyster does. We need to feed it. I used ponds 300 metres in length. This opened into a swamp area, but the water quality was very good coming in. It was fairly clean and had no excess of nutrients, was relatively low in the density of algae. So I added the fertiliser to there to spike the culture and just let nature take care of itself. So everything was in balance beforehand, everything roughly stays in balance. All you're doing is increasing the potential of the crop, the productivity of a given pond area. There's a great book, there's several great books by Boyd to look into the addition of fertilisers and stabilisers within pond culture to improve the dynamics of any given waterway. And that's exactly what we're doing here, is improving the dynamics of it. We're increasing the concentration of algae. The water goes slightly green, it might go brown, it might have a yellowish tinge. It doesn't really matter. It's the overall food value of a bell curve of species. Not one green algae, not one diatom, not one blue-green algae, but a bell curve of natural culture. So all we do is fertilise this. This then occurs. We monitor that under a microscope, so we have some idea what's going on. But basically, it'll look after its own health. So we take that flow rate and that food quantity and we put it through our oysters. And then we just sit back and watch them grow.